Well, good morning. I have a couple of announcements. I seem to be quite loud this morning, so maybe we could tune that down a bit at the back. Your exams can get picked up in discussion this week. And is there anybody who didn't get breakfast this morning? I saw a hand right over there. Okay, could you pass that back to that drooling person back there? And I'm sad to say that now for 5 times in a row, every single first hand I have seen has been from a woman. So I can either deduce that all of the guys are so sharp and so with it that they don't need anything, or the other hypothesis may be true. So once and only once this semester, this is affirmative action for all the drowsy males out there. You need a coffee. Okay. All right. Last time you guys are getting the benefit of the doubt, folks. I'm going to hand that back to that sleeping gentleman. Thank you. Okay, I sent out in BSpace last night a couple of attachments that I know that some of you had trouble finding them. Did anybody find the attachments on BSpace? Yes? Did anybody, could some one person who found them identify yourself? You found the attachments on BSpace. Where did you find them? In the messages. Okay. So the attachments, the way I send them out typically is like that, so look for them in the messages, and I'll have my assistant post them also in my folder today. So that was about 30 of the emails that I got last evening. The other 30 of them were generally of the type, do we have to know this? Are we responsible for that? Well, there are several answers to that question. The fundamental answer is no. You're not responsible for anything you don't want to know. There's all kinds of careers that one could choose. But you chose this one. You chose to come to Berkeley. You chose this course. You're choosing a major for which this course is relevant. You have 168 hours in each of your weeks. We are together for 2.5 hours. So with respect to the question, is what you talk about in class important? Well, you figure it out. If I'm going to spend the time on it, what do you think? Okay? So no more answers to questions of that type. I will simply ignore them. You will also find that the probability of me responding to your emails late Sunday night is inversely proportional to the length of the response that would be required. So if you ask me, could you explain mitosis? You won't get an email explaining mitosis. Okay. So what I want to do, and before I forget, let me alert you to a fantastic resource that is provided to us free of charge by the American Society of Cell Biology. So their website, www.ascb.org, near the bottom have links to what are called iBio seminars. This is the brainchild of Ron Vail, one of our colleagues at UCSF, a great cell biologist. And what these are very high quality recordings, much higher quality recordings than the webcasts from this class, of individuals talking about different topics on which their research is focused. Now, on average, the level of detail in these lectures is well beyond that which one would expect you to master in an introductory biology class, but these are fantastic lecturers, and some of these are of particular relevance to the material that we'll discuss in class. So Dave Morgan's lecture on cell cycle control will be particularly relevant to our discussion next time. That next lecture will probably be the most challenging lecture for most of you in the entire BIO101a series, so I'll warn you about that. Elizabeth Blackburn, who used to be one of our colleagues here at Berkeley, has a lecture that's relevant to lecture February 27th, and also to one of the problems on the thought questions that I handed out for Friday's lecture. Ava Nagala's lecture will be relevant to the issues we'll talk about today, as will Dick McIntosh's. Ron Vail's will have a slight relevance to the motors that we'll talk about at the end, and Julie Theriot's lecture will be relevant to many of the topics that we'll discuss in the first weeks of this class, or this middle section of the class. So I recommend them to you. Don't get bogged down in the detail that's way beyond what we talk about in class or in the assigned readings, but they are really very valuable and extremely well done. So what we'll talk about today, as you already know from the readings that you've done, is the eukaryotic cell division process. So cell division, like DNA replication, is incredibly tightly regulated, and it must be, because most of your cells are not replicating most of the time, And if replication were ever to become unregulated, you would have problems. Cancer is sometimes described as unregulated cell growth, but in fact, it is almost perfectly regulated cell growth. Think of the following. By the time you get a tumor that's about a cubic centimeter, that tumor has been growing for a long, long time. You now know the powers of exponential growth. So if that tumor were to grow as fast per cell as a human cell can divide, about once per day, then in 10 days, the size of that tumor would be 1,000-fold bigger. In 20 days, it'd be 1,000,000 times bigger. And over the course of a year or so, that tumor would have more mass than the universe. Okay? So even tumors, which are said to be unregulated growth, are almost perfectly regulated cells. So with the study of the control of cell division is incredibly detailed and fascinating, and we'll talk about it both today and on Wednesday. Now, with respect to the division of cells, the important thing is that there is a machinery, an elaborate machinery that you'll understand in fair detail by 9 o'clock this morning, that is assembled each and every cell cycle in a transitory way. It is assembled, and then it is destroyed. That might sound kind of crazy. Why do you build something that's so important to every cell cycle and then destroy it? And the reason is that the components of this machinery are used in the rest of the cell cycle for other things. And if the assembly was left, then there wouldn't be enough material to go around. The key component of this machinery includes the spindle. You've heard about this in high school. We'll go in greater detail now. It's made of a microtubule cytoskeleton. We'll talk about microtubules at length. Centrioles, the positions at which the assembly of the spindle is nucleated, and kinetochores. Okay? This will be the machine that helps us separate the chromosomes. And there's a contractile ring then that squeezes this elongated cell in two. Then I'll not talk very much about it because your text does a pretty good job on that. So this is the machinery that's assembled and disassembled, and that machinery is regulated by a control system, which integrates signals for growth and division that each cell receives. The signals will control a cell division kinase that we'll abbreviate as CDK, and we'll talk a lot about CDKs on Wednesday, and I very strongly urge you to read the assigned readings before coming to lecture on Wednesday. Each of these kinases will have an activity that is controlled by a cyclin, a regulatory subunit of those protein kinases. And the activity of this pair and of other proteins will be controlled by a regulated destruction mechanism. So it's a preview of what's coming on Wednesday. So we'll want to really be sure to read all about that before coming to lecture, because I'll be giving you the experimental evidence for what the book describes for you. Now, as I said, cell division is highly regulated, and the range over which cell divisions are controlled is really quite astonishing. In the first few days of your life, post-fertilization and before gastrulation, which many describe as the biggest event in your life, your cells were dividing about once every 8 minutes. At this point, in your brains, the division time for most of your neurons, hopefully, is greater than once per 100 years. You don't want your neurons dividing. Those are brain tumors. Okay? So this is a huge range over which cell division is regulated. And more typical cells might be, for example, the liver cells, which typically divide once per year. A good reason, that's not so fast, a good reason not to assault your liver too much every weekend, right? And in contrast to that, the cells of the gut lining that divide about once every day because, of course, they're getting sloughed off through the material passage through your gut all the time. So each of these division times is characteristic of those cells and is highly regulated by signals from surrounding cells and from other organ systems. Questions on the outlines of cell division. This is Wednesday. This is today. Okay? Most of what we'll talk about today is going to have a nuclear focus, but let's not forget that when a cell divides, all the stuff in the cytoplasm has to also divide to both of the daughter cells, or one of those cells will be out of luck. So cell division requires partitioning of organelles and stuff within the cytoplasm. So there's three general strategies by which this is accomplished and one very special case. The three strategies are those of abundant organelles, chloroplast versus chloroplast and mitochondria, and the Golgi and the ER. So for abundant organelles like ribosomes or peroxisomes or things of that nature, the way that we achieve partitioning of organelles into both daughter cells is to simply have so many of them that any plane of cell division that cuts one cell into two will result in some of those organelles being in both of the daughter cells. That's simple. Something that's more interesting comes from the behavior of chloroplasts and mitochondria. Eukaryotic cells have a limited number of those, and each of these has been an ancient captured bacteria during the symbiosis that occurred in the evolution of eukaryotic cells. And as ancient bacteria, they retain their own individual mechanisms for fission, for splitting themselves as bacteria normally do. But of course, if you split yourself infinitely, then you end up with tiny little mitochondria or chloroplasts. That's not what's desired. And so they've actually gained a fusion mechanism for coming back together again after mitosis. So chloroplasts and mitochondria split and then come back together after mitosis, after cell division is complete. And Golgi and the ER, which are the endoplasmic reticulum, this big network of membranes, which Jennifer described for you, I think, in lecture 3, what we see in those cells is that cells prepare to go through mitosis. These organelles break up into tiny little membrane-bound vesicles, zillions of them, and these vesicles do one of two things. They either just disperse and let this mechanism achieve their partition, or they actually sometimes have mechanisms on them, motors, we'll talk about this on the last two boards, which allow them to follow microtubules into the two daughter cells. And there's examples of both. Okay? Then the final thing that I'll talk about is the centrosome, or the centriole. The centriole is really one of the most marvelously poorly understood organelles in eukaryotic cells. It's the place that the spindle grows from, and it is replicated once per cell cycle. But the miraculous thing is that in some way, the centriole templates its own duplication. You need to have a centriole to ever make a new centriole. So it is like DNA in that something about that structure allows that structure to grow a new one. And that's a really interesting problem. And one of the world's experts on that is none other than Rebecca Heald, one of the faculty here at UC Berkeley. So she's studying centriole duplication, and it's a fascinating process. Questions so far? Okay. So what I'm showing you here is now the iconic description of the eukaryotic cell cycle. And for the last 30 or 40 years, all texts have been showing it in the following way. There's a phase called G1 that used to be called the gap phase, another phase called G2 that used to be called another gap phase that separates S phase from M phase. S phase is when nuclear DNA replication occurs, and M phase is when mitosis occurs, the splitting of the nucleus, and at the end of mitosis, cytokinesis. Now, what we recognize more now is that G1 and G0 really are not so much gaps as when things don't happen, as they are periods of time during which decision-making, molecular decision-making occurs. G1 is sort of the executive phase and deals with how a cell asks itself, am I big enough to divide? Because, of course, cells have to be big enough to divide, otherwise they get smaller and smaller and smaller and smaller and go into mitotic oblivion, right? So the cell says, am I big enough? Are there sufficient nutrients to get me through this next cell cycle? It turns out, when times are bad, eukaryotic cells want to be in G1 or G0, and we'll get to G0, and it makes the decision to leave G1 to start S if and only if there is enough nutrient to get it through that cell cycle. And then the other thing it does is to respond to various factors from other cells that inform it about its environment. And these growth factors, some of them are positive regulatory factors, and some of them are negative regulatory factors, like platelet-derived growth factor, or epigen, erythropoietin growth factor, or various other growth factors that have been turned into pharmaceutical drugs. Now, if the decision is not to go forward, as in most of your cells, the cells actually leave the cell cycle and go into a state called G0, which really is more or less the resting state. They still metabolize. They still carry out all of the processes that those cells ordinarily are designed to do for their specialized type, but they're simply not engaged in making more of themselves. Now, S phase is highly variable in its length. It's shown here as an equal fraction of the cell cycle, but that's just to represent for cartoon form. S phase varies quite a lot from organism to organism. It depends in part on the genome size. Something you should know, though, is that it is the nuclear DNA that replicates in S phase. Mitochondrial and chloroplast DNA goes on replicating all the time throughout the cell cycle. G2, we now recognize, is another decision-making point, and in G2 is when the cell evaluates whether every single base in its genome has been replicated or not. If it has not, there is a mechanism that causes the cells to halt here in G2 until that last nucleotide gets replicated. So if you damage DNA or you limit nucleotides, the length of S phase just continues into G2. G2 halts progress until the cell has a surveillance mechanism that says, yes, my last nucleotide is replicated. There is another so-called checkpoint here in G2 that asks, has each kinetochore achieved bipolar attachment to the spindle? This is really a process that it happens in mitosis, but the evaluation mechanism of mitosis and the end of G2 are kind of overlapping. There are not clear boundaries between exactly when the surveillance mechanisms are continuing into mitosis versus staying in G2 end. So don't get hung up about that. And finally, M phase, of course, is when mitosis happens. You've learned about it in high school. The text has excellent descriptions and pictures of it. I'll discuss a subset of that as we go on today. Now, in the nucleus, of course, what we're most focused on are the chromosomes. And here is a representation of a chromosome that allows me just to make a few points. First of all, in each lecture, what a line represents is variable, right? Sometimes a line is a single strand of DNA, sometimes a line is a double strand of DNA, sometimes it's a transcript. Today, a line will be a single strand of DNA and all of the proteins associated with that DNA, and that's a chromosome. A chromosome consists of a single DNA molecule stretching from one end to another. We didn't actually know that until relatively recently, that it was one continuous DNA molecule. There is a position on a chromosome called the centromere. The centromere you can think of as a point, and that is the point at which a protein assembly occurs called the kinetochore. So the kinetochore is the series of proteins that bind to, recognize, and assemble into some structure called a kinetochore at the centromere. And it is the proteins of the kinetochore at the centromere that allow chromosomes to attach to the spindle. Okay? Telomeres are the ends of chromosomes, and one of the thought questions that I gave for you today is to think about whether or not the DNA replication mechanism that we discussed last Friday is adequate to actually replicate the very ends of chromosomes, telomeres. You should be able to deduce that, in fact, what I described for you would be good at replicating most of the chromosome, but it couldn't possibly replicate the very end of the chromosome following the rules that we learned about DNA polymerases. So without some special mechanism, our chromosomes would get shorter each and every cell division, and eventually we wouldn't be here. So the replication of telomeres was a problem that was solved in Stanley Hall by Carol Greider, a Berkeley student, and Liz Blackburn, who got the Nobel Prize for this last fall. They discovered a specialized DNA polymerase called telomerase, And the role of telomerase is to synthesize telomeres in a fantastically interesting way that solves this end replication problem. And Liz talks about that in her lecture on the American Society of Cell Biology website. So if you want to learn more about telomerase, look at the first of the 3 sections on Liz's lecture. Okay. The other things that chromosomes have, are origins of replication. We talked about how E. coli has a single origin of replication. All eukaryotic chromosomes that I know about have multiple origins of replication. In fact, the number of origins of replication is a function of when in development we look. Early in development, the very rapid cell cycles require many different origins of replication. As cell division slows down, they actually use fewer and fewer origins of replication. So this, too, is another regulated step in cell division, whether a particular origin of replication is active or not. So many, many regulated steps here. Any questions on that? Really? Okay. We'll move into an area now that is really, really simple, yet for reasons that baffle me, tends to be a little bit confusing from year to year. So if you're not confused, great. If you have any questions on this board, let's take care of them now so that you don't carry that confusion forward. So we'll talk about chromosomes. As I said, bacteria typically have one chromosome and it's a circle, and they have plasmids. What's the difference between a plasmid and a chromosome? Well, generally, plasmids have genes that are not absolutely essential for the life of a bacteria, but a chromosome is essential for the life of bacteria. So a plasmid is an extra little mini chromosome that carries accessory factors that are useful, like drug resistance elements. Eukaryotes typically don't have plasmids, but there are exceptions to that. And typically, they have multiple different linear chromosomes. Why they have linear chromosomes and not circular chromosomes should become clear to you after you understand recombination. And you might want to make a note to yourself to try to understand why eukaryotic chromosomes have to be linear, and ask yourself that question 2 weeks from now when you'll be prepared to answer it. I have to introduce 2 new vocabulary words here. One is haploid, which is the word that is used to describe either organisms or the word haploid refers to one set of chromosomes, which means one copy of each chromosome. And diploid refers to organisms or cells or circumstances in which we're dealing with 2 sets of chromosomes, 2 copies of each. So each of you, I'm happy to say, is undoubtedly a diploid organism, and some of your cells are haploid cells, the gametes, that are providing one copy of each of the chromosomes, so one set of chromosomes. Diploid is 2 sets of chromosomes. So you're most familiar with diploid organisms, but in fact, many of you have come in contact with other ploidies relatively recently. Anybody had a banana for breakfast? Yeah, had a banana? You know what the ploidy of the banana was? Triploid. Triploids have 3 copies of each of the chromosomes. And did you break a tooth when you were eating that banana? Right. The reason that you don't, you'll understand after our discussion on meiosis. Triploid cells, when they go through meiosis, form small, tiny, little, pathetic aborted seeds because of problems with triploid meioses, and that's why you didn't break a tooth while biting through that. Watermelons are tetraploid. Certain frogs are tetraploid. There's certain plants that become hexaploid. Wheat became hexaploid about 10,000 years ago, which sort of allowed civilization to happen. So ploidy is quite variable among species, between species, but within a species, all individuals of a species maintains that ploidy. So we're going to use a nomenclature here called N to represent the number of chromosomes per haploid set. You'll see why this becomes useful in a minute. So for humans, N equals 23, right? That's the number of different chromosomes in each of your gamete cells. But because you're diploid cells have 2n chromosomes. So each of your cells, your somatic cells, have 46 chromosomes. Now, the number of chromosomes in a haploid set, or the total number of chromosomes in 2n cells, varies a lot from organism to organism. There's nothing special about 46 for the diploid number. We share that number with potatoes. Okay? There's lots of animals, lots of plants that have more different chromosomes than we do. For example, N is 3 in Schizosaccharomyces pombe, a yeast, and in the Indian monkjack, a deer. Okay? So there is no relationship at all between the number of chromosomes and the biological complexity of that organism. There is even an exotic example of an ant in Australia, the Australian jack jumper ant, which is one of only 2 organisms that I know about in the biosphere in which N equals 1. It has all of its genome on a single linear chromosome. And in ants, as in social insects, sex is determined by the number of chromosome sets. So males are haploid. They have 1 copy of this 1 chromosome, and females have 2 copies of this 1 chromosome. If anyone can find for me a eukaryotic organism that actually has 2 chromosomes, I would love to know about that. I have searched for that and have never found one, but if any of you do, please let me know. I'll be most appreciative. So the number of chromosomes that an organism can have can really go from 1 to very large numbers and has got no relationship to its complexity. But the amount of DNA in a nucleus does not determine its complexity, but is more closely related to the complexity of an individual. So for example, humans have 3 times 10 to the 9th base pairs in its 23 haploid chromosome set. A different yeast has 12 times 10 to the 6th in its 16 chromosome haploid chromosome set. So the amount of DNA in a species is really quite variable. And we use a different value, the so-called C value, to represent the amount of DNA in an individual, and N to represent the number of different chromosomes in an individual. And you'll see why we want to have both of these measures at our disposal in just a minute. So any confusion so far? Think about it. You shouldn't be confused, but from year to year, people tend to get confused. Yeah, what's your question?
For telomeres, like the cell in like the gut lining, is that one of the reasons like we can't live for a really long time? Because eventually it's going to run out, and then you're not going to be replicating like actual useful DNA anymore?
Yeah, great question. What's your name? Harash. Harash asked the question about telomeres. Is the reason that we can't live forever the fact that we can't continue to replicate our telomeres? That appears to be the answer for certain cells in culture. So when you take a human cell and put it in culture, there's a certain number of cell divisions that it can go through, and then it just craps out. It goes through something called crisis, and then it undergoes a death, a hideous death referred to as apoptosis. I think Gary will talk about that later. And those cells in culture don't have their own telomerase. If you force them to have telomerase, they can continue to live. So for human cells under specialized conditions in the lab, telomerase is limiting for longevity. That seems not to be what's limiting for longevity, and I think in my last lecture we'll return to the issue of longevity and discuss what actually is limiting for longevity for the organism as a whole. Great question. Other questions about this? Yes?
How would I find the C value with an equation?
I'm so glad you asked me that question. We'll get there, okay? And we'll get there in the context of the relative C value rather than the absolute C value. To get the absolute C value, all you really need to do is to know the number of cells that you start with, you purify all of the DNA, and then you simply measure how much DNA is there in that number of cells and divide by the number of cells. Okay? And there's chemical assays for the amount of DNA that's really very straightforward. But what we're going to do now is to talk about the measurement of N and the measurement of C as it's applied to useful experiments and questions that we want to answer. So how do we measure N and C? Well, to measure N, it's really quite easy. We simply arrest cells in M phase with a drug. And in M phase, the chromosomes are all highly, highly condensed. And here's an interesting calculation for you to do. Your genome is approximately 1 meter long and 10 angstroms across, laid end to end. It fits in a nucleus, which in many cell types is about 5 microns across it. It's a fun arithmetic to figure out how much condensation has to happen in your genome to be able to fit in that small space. The highly condensed state of chromosomes, though, allow us to stain them with a stain that stains DNA and simply look in a light microscope at about 400x. You can see them at a lower power, but about 400x is a pretty good way of getting a reliable count on the number of chromosomes that you have. Any of you that have sort of a prenatal diagnosis at some point will have a nice picture of your fetus's collection of chromosomes provided to you. One of my friends has his son's karyotype, his son's picture of his chromosomes as his screensaver. It's kind of cute, okay? At least it was until his son became a teenager. Then it'd be, oh, yeah. Now, what about C? Well, we're going to measure C not chemically by counting the number of base pairs. We're going to count C as a function of when cells are in the cell cycle, because to understand the control of the cell cycle, we need to be able to measure where the cells are in the cell cycle. So here's how we do it. We take a collection of cells that we're interested in, and we stain them with a fluorescent dye that binds to DNA and to nothing else, and the binding of this DNA, the fluorescent signal is proportional to the amount of dye that is bound to DNA. Linearly proportional, okay? So then we'll use an instrument called a FACS, a fluorescence-activated cell sorter, or more commonly used in many procedures, a fluorescence-activated cell scanner that scans cells without sorting them. And here's what we do. We simply allow a stream of cells to go between a laser and a photomultiplier tube, a detector of some point. We tune the laser so that it is optimized for the excitation wavelength of the dye, and we tune the detector so it is optimized for the emission wavelength of the dye. So the amount of signal that that cell produces is a function of how much dye is bound in its nucleus, which is a function of how much DNA it has, which is a function of where in the cell cycle the cell is. This detector runs through electronics to a computer, which can then take an orifice through which the cells will flow and adjust the charge on one side or the other of the orifice. Now, you know that cells have an elaborate collection of molecules on their surface, sulfonated proteolipids and this or that provides a particular charge to them. So depending upon what the detector sees for that cell, it can cause the cell to get deflected either this way or that way. So you can actually purify tubes of cells at different points in the cell cycle. Or you can simply take a population of cells and in 1 minute measure for about 1 million cells where in the cell cycle that population of cells are. So let's imagine now a plot of the number of cells versus the fluorescent signal increasing in that direction. And here is the result that we might obtain from a culture. Okay? So if this were a culture of your cells, you in the end, what's your name? Grayson. If this were your cells, culture of your cells, tell me what you would deduce about that culture of your cells. What could you learn? Okay, where are these guys? Yeah, these would be the cells that are in G1 or G0. And these would be the cells that, of Grayson cells that are either in G2, preparing to go into M, or actually in M. What about this dip right here? Well, 3C would be right here. Would you want one of your cells to go into mitosis before it had replicated all of its DNA? Yeah, these would be the cells that are in S phase. So Grayson's got it exactly right. Here's S phase. Okay? So it's a really powerful tool. And it's amazing how many different exam questions can be structured by looking at different kinds of outputs from a FACS scan on cells treated in different ways. So let's make sure that the concept behind a FACS scan is solid. Any questions about that? It's not complicated. The gizmo is quite complicated. Herzenberg spent years figuring out how to build one. Yes, sir, what's your name? Yishi.
What is the plate, charge plate?
These plates are simply a way of deflecting cells into one collection vessel or another, depending upon how much DNA the cell has. So it's a way of sorting cells. Yeah, you can get the graph either. The graph actually comes right off of a computer right here. Whether you sort or not, you get the graph. The machine is sophisticated, also allows you to sort. There was a question over here someplace? Okay, good. Okay, so I'm not going to, yes, question.
In the graph you mentioned, G0, G1, M, and X. What about G2? What would it be in the graph?
Yeah, a G2, what do you think? So here's G1, right? This is not 2C and it's not 4C here, right? So that's got to be S. How much DNA is there in a G2 cell with respect to C? Mm-hmm. So G2, M, all those cells would be in this peak. Yep, good question, thank you. Okay, yes? Sure. Oh, great question, yeah. It depends upon what you're trying to, well, the dye stains DNA. Now, whether you can see that in a light microscope or not depends upon its condensation. The laser and the photomultiplier tube can see that fluorescence regardless of whether it's condensed or not. Thanks. Okay. So you've been through mitosis already lots of times, high school biology, AP biology. The text is great. I'm not going to lecture the details about mitosis. But what I am going to try to do is to prepare you not to blow your minds in meiosis. Meiosis, I know you think you know it, but I know you don't. And I know you won't really understand it unless you really understand just a couple things about mitosis. So I am going to belabor them to death. Okay? So let us consider now an example you carry out, the Indian monkjack. They're kind of cute. They're about that big. I hear they're tasty, though I've never tried one. A vegetarian in the crowd. Here is a nucleus, and what I've represented are the 2 copies each of the 3 different chromosomes that are in a haploid set of the Indian monkjack DNA. There's 2 purple ones, 2 blue ones, and 2 green ones. And as you know, one of the green ones came from the Pa, and one came from Ma, one of the blue ones, and so on and so forth. Now, the 2 copies of each of these chromosomes, the pair, is called a homologous pair of chromosomes. Homologous meaning they're both the same number. They're both the green one, right? Homologous does not mean identical. It means they're pretty close, but they're not identical. You know that because none of you look exactly like Mom or exactly like Dad. They are very, very similar, but they carry the differences that came down through the ancestral line of the mother and the father monkjack deer. Okay. So, quick quiz. How many DNA molecules are in this cell? You can't answer that question because I didn't tell you about how many mitochondria there are. How many DNA molecules are in this nucleus? 6. Very good. What is N? N is 3. Yep. What is C, relatively speaking? 6. Your options are 2C or 4C. This cell, 2C, exactly. How many chromosomes? 6, very good. How many homologous chromosome pairs? Oh, that was great. Well done. Okay. Now I got to warn you, something's going to happen here during S phase. And that is, we are going to change, damn it, the definition of a chromosome. I can't help it. I didn't make it up. This, it was way before me. So we've gone through S phase, and now look. Each of the lines that was an entire chromosome before is now 2 lines. Okay? These 2 lines represent the sister chromatids, which make up a chromosome, that were produced by semi-conservative replication that we learned about last time. Okay? And the same thing happens to all of the chromosomes. So we still have 6 chromosomes. We still have 3 pairs of homologous chromosomes, but each of those chromosomes now is a pair of sister chromatids. Any questions about that? It is an ambiguity that biologists have had to live with for decades, and I apologize for that, though it's not my fault. Good, I'm glad you've got that. It's really pretty straightforward. So now, if I were to ask you how many DNA molecules there are in this nucleus, you would say, DNA molecules, 12. Exactly. Okay. So what is it about these chromosomes? Well, what's cool about these chromosomes is that they are held together along their entire length in a process called cohesion by proteins named creatively cohesins. And it turns out that the world's expert on cohesins and sister chromatid cohesion is none other than Doug Koshland, one of our colleagues here at Berkeley, whose lab is in Barker Hall, not Koshland Hall. How did that happen? Each of these sister chromatids has started to assemble a kinetochore, a collection of about 50 different proteins that assemble at the centromere and connect that centromere to the spindle. We'll get to the spindle. And it just so happens that Georgiana Barnes, one of the world's experts on kinetochore structure, is also one of our colleagues here at Berkeley, also in Barker Hall. So we're early in M phase here. The chromosomes have not yet even really condensed, and they are scattered through, they're sort of not aligned in any particular way, but note that the nuclear envelope has broken down, as it does in many eukaryotes, but not all eukaryotes. This seems crazy to me, right? You've got the crown jewels protected in this nucleus, so why in mitosis do you dissolve the nucleus? Whew. Go figure. Yeast does not do that. Human cells do. Another example of yeast being more highly evolved than human cells. Okay. Now, you all know that this is a representation of metaphase. I know you know that. But here's what you'll get confused by. If I were to ask you to describe the pairing of homologous chromosomes in mitosis, you should go, no! They don't pair, okay? You will be confused because if you look at these 2 lines here, it looks like there's a pair of something. You will think that's the pairing of homologous chromosomes. It's not. In mitosis, homologous chromosomes never, ever pair. At least until you get to MCB 140, and then it's just for a little millisecond, okay? Okay? These are the sister chromatids that are still cohesed. Now, what's happened here in metaphase? Well, we have assembled a spindle. We have a centriole here and here that have assembled a spindle by seeding the growth of microtubules, protein tubes, literally tubes that grow from the kinetochore the centriole, out and attach to the 2 kinetochores for each pair of sister chromatids. Okay? You've seen that before. So let me give you a little more information, though, than you've seen before. Microtubules have a polarity to them. They're arbitrarily called minus ends and plus ends. The growth of a microtubule occurs only at its plus end, not at its minus end. So all of these microtubules have the same orientation in the cell. Microtubules are made of polymers of alpha and beta tubulin, which looks in reality not very much like what I tried to draw here, but described beautifully in an animation in Ava Nogales' lecture on the ASCB website. Probably one of the best animations I've ever seen in biology. So this is the plus end, and the plus end of a microtubule is made up of tubulin that has locked in its embrace GTP, ribo-GTP, a source of energy. At the minus end, GTP is depleted. The microtubules down here have GDP. And it is only the microtubules that have bound to GTP that allow other microtubules to attach, other tubulin subunits to attach to it. That's why growth always occurs at the plus end. That's where the GTP is. The GTP puts the tubulin in a configuration to allow growth. So the microtubules then have attached to all of the kinetochores, but there is a surveillance mechanism going on. Each of the kinetochores has to be attached to a microtubule. Each pair of sister chromatids has to be attached to 2 different microtubules from 2 different sides. It doesn't work if 2 microtubules from the same side attach to the 2 kinetochores on the same sister chromatid. It is only when the opposing forces align that the cell can detect that tension exists on each of these kinetochores, and at that point there is an elaborate cascade of protein proteolysis that leads to the destruction of Doug's cohesins. So at that point, the glue that is holding the chromatids together along their entire length dissolves. Okay? So that's good. We have to dissolve that glue to separate the chromosomes. And now what you're familiar with is the later phases of mitosis. And the chromosomes seem to move toward the poles, allowing cell division to occur between that. But there's some tricks here. We know there are microtubules that make up the spindle that don't attach to kinetochores. What's that all about? The other question is, where does the force come from to move chromosomes along the spindle? And amazingly, in March of 2010, we don't have satisfactory answers to those questions. I'll tell you what we know. I might even tell you a little more than we know, but that's okay. So we'll deal with what moves chromosomes on the spindle and which end of the microtubule shrinks. So those are the last 2 questions we'll deal with today. Here's a slightly better diagram of a microtubule. What I'm showing you is a sketch of a motor protein. There are proteins in cells. There's about at least 8 of them that work on the spindle that actually use the energy of chemical hydrolysis of ATP to move things. One end literally walks, and one of the movies by Ron Vale will show you a good model of that, and the other end attaches to cargo. And motors can have 2 different orientations. There are plus end directed motors that walk this way, and there are minus end directed motors that walk that way under microtubules. So far, no one has ever found a motor protein with a transmission. It is always going to the plus end or it is going to the minus end, depending upon the type of motor that it is. So imagine now, how are we going to spread the spindle? Well, we can spread the spindle by having a plus end motor with its cargo attached to one microtubule, walking towards the plus end of the other overlapping microtubule. This will actually force the elongation of the spindle, which you will see in the movies and in your text, occurs as in telophase, the cell elongates. But the question is, what brings the chromosomes to the end of the microtubules to align in metaphase, and then gets them to go in that direction after the cohesins have dissolved? And frustratingly, we don't really know. Here's what we do know. In the kinetochore, there's 50 different proteins, including motor proteins. So there is the potential for motor proteins to actually walk towards the minus end of microtubules to move chromosomes during mitosis. But the other thing that happens at that kinetochore is that there is a ring of proteins called the dam ring, not the dam ring, the dam ring. And this makes a ring of proteins around the end of the microtubule that connects the chromosome to that microtubule and may, according to Barnes and Nogales, may actually control the stability of this plus end. So imagine what happens suddenly if that plus end of the microtubule chews up all of its GTP. Well, if it does, it becomes unstable, and it starts to peel apart like a banana. The subunits all dissociate, and what do we get? Entropy. So there's a whole school of thought that what actually drives the chromosome to the minus end is the entropic drive that comes from the disassembly of the microtubule, with this ring surfing the wave ahead of that unpeeled microtubule, drawing the chromosome to the other end. And I highly recommend Eva Nogales' video to show you her model of how that'll work. Okay? So be sure to read about the regulation of the cell cycle by Wednesday. If you don't, all hope will be lost. And I'll see you then. I have office hours today. I think it's from 12 to 1, someplace that's described on the website.